Here is a combined summary of the 2 pages. Brain network analysis is crucial for understanding neural interactions and identifying potential biomarkers for clinical phenotypes. However, brain signals such as blood oxygen level dependent, bold, time series generated from functional magnetic resonance imaging, fMRI, often manifest 3 challenges: missing values, irregular samples, and sampling misalignment. These challenges impact downstream brain network analysis and clinical outcome predictions. To address these challenges, this work proposes a novel model called BrainODE, which achieves continuous modeling of dynamic brain signals using ordinary differential equations, ODE. By learning latent initial values and neural ODE functions from a regular time series, BrainODE effectively reconstructs brain signals at any time point, mitigating the 3 data challenges altogether. The proposed model utilizes graph convolutional networks, GCNs, on 2 graphs, capturing structural, spatial, and functional, temporal, ROI relations in brain networks. These graphs not only leverage common wisdom about brain connectivities but also provide opportunities for deriving clinically actionable discoveries. By mitigating the impact of low data quality, BrainODE enhances the usability of dynamic brain signals in clinical predictions with improved performance for various downstream brain network analysis models. The key contributions of this work include recognizing the importance of reprocessing dynamic signals in brain network analysis, identifying 3 major data challenges, and providing a unified framework to address these challenges through continuously modeling interactive brain signals. The proposed BrainODE model utilizes spatial and temporal graphs to capture complex interactions among ROIs, facilitating continuous neural ODE learning and inference. Experimental results on 2 real-world datasets demonstrate the effectiveness of the proposed model, with significant performance improvements in classification tasks. The model's efficacy in individually solving the 3 aforementioned data challenges with superior performance is also demonstrated. This work has implications for brain analysis and potentially searching for associations between brain signals and clinical outcomes such as mental disorders. Here is a combined summary of pages 3 and 4 of the research paper. The paper proposes a novel approach, BrainODE, for continuously modeling dynamic brain signals. The framework consists of 3 key components: brain latent state learning, brain temporal relation and spatial relation incorporation, and autoencoder-based end-to-end training. Brain latent state learning involves capturing the complex relationships among ROIs and integrating them with neural ODE. This is achieved through a CNN-based encoder to capture brain activity and a self-attention module to model long-term dependencies among dynamic signals. The CNN-based encoder captures intricate features from grid-like data, and the self-attention mechanism captures dependencies and relationships between entries across a wide interval of time steps. Brain temporal relation and spatial relation incorporation involves constructing temporal and spatial graphs to represent the multifaceted relations among ROIs. The temporal graph captures correlations among dynamic brain signals, while the spatial graph encodes structural connectivity of ROIs based on their 3D coordinates. These graphs are used to enhance the learning of the neural ODE model through additional encodings of the learned initial values. The learned initial values are then refined through 2 distinct GCN layers, which learn the spatial and temporal relations among ROI channels, aiding in the derivation of initial states for ODE inference. An ODE solver and decoder are utilized to obtain the brain signals at desired regular time steps, which can lead to enhanced performance and subject classification. The proposed approach addresses the challenges of missing values, irregular samples, and sampling misalignment in dynamic brain signals. By leveraging the strengths of neural ODE and graph convolutional networks, BrainODE provides a novel solution for continuous modeling of dynamic brain signals, which can lead to improved brain network analysis and downstream predictions. In this research paper, the authors introduce a novel approach to reprocessing dynamic brain signals for improving the performance of various base models in functional brain network analysis. The proposed model, BrainODE, integrates spatial and temporal relationships between regions of interest, ROIs, in the brain, utilizing a graph neural network, GNN, for spatial relations and a continuous time ordinary differential equation, ODE, for temporal relations. The model is trained in an end-to-end fashion with a mean squared error, MSE, loss for signal reconstruction, and a Kullback-Leibler (KL) divergence term for normalizing the initial states of the ODE. The authors evaluate the effectiveness of BrainODE by comparing it with three opponent models: polynomial interpolation, poly, recurrent neural network, RNN, and TST+, TSTP, on two real-world datasets: Autism Brain Imaging Data Exchange, ABIDE, and Adolescent Brain Cognitive Development Study, ABCD. These datasets are used for diagnosing autism spectrum disorder, ASD, and predicting biological gender, respectively. The results show that BrainODE significantly improves the performance of base models by addressing missing values, irregular samples, and sampling misalignment in the brain signals. The average area under the receiver operating characteristic curve, AUC, and accuracy, ACC, improved by 27.4% and 16.9% on ABIDE, and by 15.6% and 8.0% on ABCD, respectively. The paper also conducts an in-depth analysis on addressing missing values and irregular samples, and evaluates the impact of individual components and hyperparameters of BrainODE. It demonstrates that BrainODE outperforms its opponents in both interpolation and extrapolation settings, indicating its ability to effectively handle missing values and misaligned samples in brain signals. In the proposed study, the authors introduce BrainODE, a novel deep learning model that utilizes ordinary differential equations, ODEs, to analyze and predict dynamic brain signals from functional magnetic resonance imaging, fMRI, data. This approach addresses missing values in brain imaging data by modeling the temporal and spatial relations among regions of interest, ROIs, using a graph neural network framework. The results demonstrate BrainODE's superior performance in interpolating and extrapolating missing data points, outperforming other state-of-the-art methods. The authors also conducted an ablation study to understand the contributions of different components in BrainODE. Removing the position encoder, temporal graph, or spatial graph resulted in varying degrees of performance decline, highlighting the importance of each component. Additionally, a hyperparameter analysis showed that increasing the latent embedding size and kernel size can significantly improve the model's performance, with optimal performance achieved within a specific range of these parameters. The study also explored addressing irregular samples and sampling misalignment, which are common issues in fMRI data. BrainODE consistently outperformed other methods under these conditions, demonstrating its robustness and adaptability. The paper concludes by discussing limitations and potential future directions, including the incorporation of real graphs such as structural brain networks constructed from diffusion tensor imaging, DTI. The authors also note that the current study is focused on generating synthetic data, and future work could involve applying BrainODE to real-world clinical applications. The paper discusses the implementation of a novel framework, BrainODE, which integrates brain imaging data with graph neural networks and ordinary differential equations to model dynamic brain networks. It leverages the strengths of both domains to provide a comprehensive understanding of brain function and structure. The proposed method is evaluated on two datasets: the multi-site Autism Brain Imaging Data Exchange, ABIDE, dataset and the Adolescent Brain Cognitive Development, ABCD, study. The BrainODE framework consists of several key components. First, it uses a short-term encoder, a long-term encoder, and a decoder to learn and generate brain signals. The short-term encoder is a single-layer CNN network with a filter size of 4 and an added ReLU nonlinearity. The long-term encoder utilizes self-attention mechanisms with a single attention head and positional encoding. The framework also incorporates a generative model that uses a fourth-order Runge-Kutta method from the TorchDefect package as the ODE solver. This allows for the generation of subsequent ROI signals based on the initial latent states. The model is trained in an autoencoder-based framework with an end-to-end fashion, using a mean squared error, MSE, loss and a KL divergence term to normalize the initial states of the ODE. The model is trained for 50 epochs, and the test result of the best validated value is reported. In terms of parameter tuning, the paper follows the implementations from the authors of the Brain Network Transformer for baselines such as FBNet Gen, BrainNet CNN, and Vanilla TF. A grid search is used to tune key hyperparameters based on the provided optimal configurations. For FBNet-Gen, different encoders, 1D-CNN, grew, with varying hidden dimensions, 8, 12, 16, are explored. For BrainNet-CNN, different dropout rates, 0.3, 0.5, 0.7, are tested. Similarly, for Vanilla-TF, the authors search for the optimal configuration. The paper also provides a comprehensive list of references, detailing various studies and techniques used in the development of the BrainODE framework. These include works on graph neural networks, time series forecasting, and brain network modeling. The use of these references demonstrates the interdisciplinary nature of the research, combining insights from machine learning, neuroscience, and computer science. The research paper presents a comprehensive analysis of various methods for addressing missing values in time series data from brain imaging studies. It introduces BrainODE, a novel approach that leverages transformer layers and graph neural networks to model complex relationships between different regions of interest, ROIs, in the brain. The authors compare their method with existing techniques such as polynomial interpolation, RNN, and TSTP using two datasets, ABIDE and ABCD. The results are presented in Table 4, which includes standard deviations for each method. BrainODE outperforms other methods in terms of average root mean squared error, RMSE, demonstrating its superior capability in modeling ROI relations. Ablation studies reveal the importance of the position encoder, temporal graph, and spatial graph components in BrainODE's performance. Removing these components leads to significant increases in standard deviations, highlighting their importance. The authors also provide details on the software versions used for BrainODE and brain network analysis in Table 5. This information is crucial for replicating the experiments and understanding the technical requirements for implementing BrainODE. In conclusion, the paper showcases the effectiveness of BrainODE in handling missing values in time series data from brain imaging studies. By leveraging transformer layers and graph neural networks, BrainODE improves upon existing methods and provides a more stable and accurate modeling of complex ROI relations.